DNA replication is the process of producing two identical replicas of DNA from one original DNA molecule. The method used for copying DNA replication is semi-conservative replication because half of the original molecule is kept in each of the new molecules. DNA replication takes place when a cell is not dividing, but the cell is in interphase, more specifically S phase. In this slide, you can see the DNA molecule with the nucleotides inside. These nucleotides are adenine, guanine, cytosine, and thymine. These nucleotides fit together like a puzzle by the hydrogen bonding in between adenine and thymine and guanine and cytosine, with adenine always going with thymine and cytosine always going with guanine. Adenine and guanine are purines, whereas cytosine and thymine are pyrimidines. Nucleotides are made up of a nitrogen-containing base, a pentose sugar, and a phosphate group. DNA is made up of deoxyribose sugar and a phosphate group, which make the backbone of the DNA. The DNA molecule has two polynucleotide chains, which are held together by the bases of the two chains. Scientists Watson and Crick demonstrated that this could be done by twisting the DNA into a helical shape and running the strands in opposite directions. This gives DNA the name double helix. The 5' and 3' of the DNA molecules are represented by the location of carbon atoms on the sugar. The two ends of a DNA strand are called the 5' end and the 3' end, but the 5' end is a phosphate and the 3' end is a sugar. However, the entire backbone of the strand is made up of a phosphate and sugars bonded together. DNA helicase is known as the unzipping enzyme because of its role in breaking down the hydrogen bonds between the nucleotides and separating the DNA. The top strand of DNA becomes the leading strand, and the bottom strand of DNA becomes the lagging strand. The leading strand runs from 5' to 3', and the lagging strand runs from 3' to 5'.
DNA primase is known as the primer for DNA synthesis to occur. Without DNA primase, DNA polymerase would not be able to add new nucleotides to the existing DNA strands. DNA primase adds a primer along the leading and lagging strands in order for DNA polymerase to come in after. DNA polymerase is known as the building enzyme. It is only able to build from 5' to 3', which makes the leading strand much easier to build because it flows 5' to 3'. This makes the lagging strand much more difficult to build because it flows from 3' to 5'. This causes DNA polymerase to have to build from the opposite direction, creating gaps between nucleotides. These fragments of nucleotides are called Okazaki fragments. DNA polymerase carries the sugar phosphate backbone with it while it is copying the DNA and putting new nucleotides in. DNA ligase is an enzyme that is introduced for the lagging strand in order to sew up the gaps in between the Okazaki fragments. Ligase does this by joining two nucleotides with covalent and phosphodiester bonds.
At the end of this process, two new DNA molecules are formed, with half of the old strand and half of the new strand each. Each chromosome is made of protein and a single long DNA molecule. This is the process in which the DNA is replicated in order for the chromosomes to be replicated for cell division. Without DNA, chromosomes would not exist.